Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog.
Hello, I'm Yvonne Stapp for Science for the Public, and I welcome you to Contemporary Science Issues and Innovations. Today, we're very honored to have with us Kim Lewis, University Distinguished Professor and Founder Director of the Antimicrobial Discovery Center at Northeastern University. Dr. Lewis is an internationally recognized expert in microbial drug resistance, and lately he's been in the news a great deal because his lab's breakthrough development of an antibiotic that seems to overcome drug-resistant bacteria. This is a very significant thing because, as you know, antibiotic resistance has led to a public health crisis. Dr. Lewis is a permanent member of the NIH Drug Discovery and Drug Resistance Studies section. He's also a fellow of the American Society of Microbiology, and he's received numerous awards and patents for his work. In addition, he's the founder of two biotech companies, Novobiotic Pharmaceuticals and Arietas Corporation. Today, Dr. Lewis will discuss his latest and possibly greatest innovation, an apparently fail-proof antibiotic called teixobactin that has caused a great deal of excitement in both the scientific world and in the media. It is a great honor to welcome Dr. Kim Lewis. Dr. Lewis, welcome.
Thank you, Yvonne. Thank you for having me. I just want to start by saying that we're, of course, excited about teixobactin, but it is a result of an effort of several teams, and probably as our conversation unfolds, I'll tell you more about
Okay, that's just fine. But can I start, though, with just getting some background? I think people are aware that we have a crisis with, the antibiotics today. Can you give us a little background about how antibiotics were discovered and what happened?
Yes, of course. The first highly successful antibiotic, as we all know, was penicillin, and it was discovered by accident. A fungus spore landed on a partially open petri dish where some staphylococcus was inoculated, and Fleming noticed that there was no growth of the staphylococcus around the fungus. Mm-hmm. So he had the foresight to conclude that the fungus was making an antimicrobial compound, and that led to the isolation of penicillin. A number of years later, in the 40s, Selman Waxman, working at Rutgers University, decided that he is going to use Fleming's discovery to systematically discover new antibiotics. So his idea was very simple. He thought that he will take soil microorganisms, and he was focusing on actinomycetes that turned out to be the most prolific producers of antibiotics, that he would take one colony of actinomycete at a time, apply it to a petri dish overlaid with the same staphylococcus or another pathogen, and look for zones of inhibition, just like Fleming did. Mm-hmm. By accident, mm-hmm. he would do it systematically. Mm-hmm. And so he did that, and that was enormously successful. He discovered streptomycin mm-hmm. and was awarded the Nobel Prize, both for streptomycin and for the method. Mm-hmm. And the method was then picked up by a number of pharmaceutical companies like Merck and Pfizer and Eli Lilly. And really what they are today is in large part can be traced to their discoveries of antibiotics that mm-hmm. transformed both them as companies and medicine as a field.
And when they did all these great and wonderful things, it looked like the cure for everything. What happened that people maybe didn't expect initially that led to the antibiotic crisis?
Right from the very beginning, it was clear to experts, maybe not necessarily to the public and to the
Right.
doctors, but to the experts, that there were some cases of resistance to penicillin, to streptomycin, and to other antibiotics as they were being discovered and introduced. But that did not seem at the time meaning the 40s and the 50s and the 60s, like a big problem, because there was plenty antibiotics that were discovered, plenty of classes of antibiotics were being discovered. And the discovery far outpaced the resistance acquisition and spread by the pathogens. So resistance seemed more like a nuisance rather than a real problem.
But then it became a real problem.
And became a problem.
Is it because of the overuse of the antibiotics? It seems pervasive now. It's in our meat. It's in everybody's taking them all the time. Was that the difficulty, or would it have been inevitable just because of the mutation rate?
Yeah, there were two different factors that contributed to our present perilous state of antibiotic resistance. So one was, as you said, Yvonne, you're absolutely right. It is the wide availability and overuse of antibiotics, using antibiotics when people really have viral infections, for example. That has been pretty standard. Or a very wide use of antibiotics as over-the-counter medications in a lot of third world countries where you do not need a prescription.
Yeah.
So that has been, of course, a major component, and then, of course, also the use of antibiotics, for growth promotion of farm animals. But apart from that, if we would have continued the discovery at the pace of the 40s and 50s, we'd probably be okay today, in spite of the misuse of antibiotics. But that was not the case. The rapid discovery and introduction of new classes abruptly ceased in the early 60s. Like somebody turned off the discovery process.
And why?
So that we can fairly confidently trace the overmining of the main source of antibiotics that Selman Waxman was working with and introduced. So it turns out that about 1% of soil microorganisms are species that we can culture in the lab easily.
Right.
And the remaining 99% of species, for a variety of reasons, do not want to grow on our petri dishes. And so if you have a limited resource of something, then of course that's going to be overmined.
Right.
And so after the 60s, people realized that they are spending their time rediscovering penicillin and streptomycin.
I see.
Instead of discovering new classes of antibiotics.
So let me get this straight. Part of this was the problem of the culturing in the petri, what could be cultured in the petri dish. This is where you come in, I think, that you turn back to where 99% of the bacteria were,
Right.
And you came up with a very different method.
So my colleague from Northeastern, Dr. Slava Epstein, who is a microbial ecologist, and I have been thinking about that problem, first independently and then jointly. So we joined forces a while ago and decided that this is a fascinating basic science problem. We were motivated just by the puzzle.
Just because of that.
At the time, I must say, of course we realized that it would be probably useful if we learn how to grow them, but our primary motivation at the time was to solve the problem.
Right. What is the problem?
Well, that is the problem. Why do bacteria not grow on our petri dishes when we seemingly add wonderful nutrients on our petri dishes?
Right.
You know, sugars and amino acids, that would be exactly what's available to them in the soil, the breakdown products of plant and animal matter. But 100 years of tinkering with conditions on a petri dish didn't allow us to move from that 1% upwards. The phenomenon itself, this inability to grow the vast majority of species on a petri dish, is known in the profession as the Great Plate Count Anomaly.
Oh.
So it's the difference between the number of bacteria that you can observe under the microscope and count them, and if you then take the same cells and put them on a petri dish, well, there's a great plate count anomaly. Only 1% of them are going to grow. And that was discovered in the 19th century by Winterberg, an Austrian microbiologist. So this is the oldest unsolved problem in microbiology.
Was it a problem, by the way, with penicillin too? They couldn't make enough of it. Did that have anything to? I didn't.
That's a different problem.
Okay.
So of course, they could grow the fungus, but in the beginning, the fungus did not want to make a lot of penicillin.
I see.
It apparently made exactly the amount the fungus needed.
I see.
Not the amount that we needed.
Exactly.
Right? We
so,
needed
right, wasn't into mass production.
vastly
right. But you were,
amounts.
yes,
Yeah.
you worked with a different situation.
Yeah, that's a different problem. And so what Slava and I came up with was a very simple, really, consideration. We realized that, there's a vast negative experience of trying to come up with a wonderful media that's going to grow everything. And we realized that that's not going to work. That's not what we want to do. We also realized that uncultured bacteria do grow in their natural environment.
Yeah.
And so that's where we are going to grow them. So then the question was coming up with a gadget to do that. And the gadget we came up with is a diffusion chamber. So the idea is very simple. You take a sample, let's say a marine sediment that we started working with, or soil, you dilute it, mix with agar, and then you sandwich it between two semi-permeable membranes. Those get glued on an O-ring, which we get in a hardware store. And so now you take this diffusion chamber and put it back, let's say, in the soil where the sample originated from. Everything diffuses through the semi-permeable membrane, so essentially we trick the bacteria. They don't know that something happened to them, right? And they of course grow and form colonies. So that was our initial big breakthrough to see that, and that for the first time gave us access to these elusive, uncultured microorganisms.
I see. When I saw the gadget that I think that you're talking about, and I will insert it in the video, I thought, that seems so obvious. Why hasn't somebody done this? Was it obvious, or did you spend a long time getting to that? Why was it so difficult?
The basic idea seemed rather obvious to us. And then we put together the first diffusion chamber, and nothing worked. Right. And then we spent quite a while, tinkering, realizing that we need to tinker with conditions, optimize it, but really nothing radical, I would say.
Yeah.
Right. So it had to be of a certain, it had to be very thin for good diffusion. The membranes themselves, the first chamber we put in the soil, bacteria and nematodes ate it up, the membrane. So because
they're membrane?
Yeah, they're made of nitrocellulose, of course. We could have imagined that.
Yes.
That will be eaten, but we didn't. So each and every problem that you can imagine, we could have encountered, we did. But then, we optimized it, and it did work. So, in a way, yes, the idea is simple and obvious, and when we tried to patent this, we were greeted with skepticism from the patent examiner, because she said this is so obvious. But so our counter argument was, well, if it's so obvious, why didn't anyone come up with it? Right. So
I'm tempted to ask that too. Why didn't somebody? It sounds like you tinkered, so I wanted to try to find out how long that took. But everybody knew about the benefits of the soil bacteria, correct?
Potential benefits.
The potential,
of those uncultured soil bacteria.
Right.
You see, I would say there are probably two different things. Well, first of all, there's a tradition, right? So in microbiology, there's a tradition. Tradition, here's a petri dish. Right. 10 bacteria, you grow them in a petri dish.
Yes,
that's
right.
like
yes,
really
that's
like
right.
ABC,
It has to work that way.
So that's one thing which is kind of hard of dispensing with. But another thing, of course, microbiologists have thought about this problem, and there was a list of reasons why, possible reasons why they don't grow in the lab, and one was really a killer. One of the reasons, potential reasons.
Yeah.
So one of the explanation was that these are simply, the vast majority of bacteria in the environment are simply extremely slow growing. So maybe they divide once a month or once a year. So you never see a colony on your petri dish. But if you wait for a couple of years, you'll see colonies. But who has a couple of years?
It's
right?
got exactly.
So essentially, the proposition was that this is a boring problem. Right? This is, first of all, a boring problem. And secondly, it will have no practical significance.
Yes, I understand.
Right?
Yeah.
And that was another barrier that I think prevented a lot of people from going there.
Right. So were you in a distinct minority then, developing this thing? I can't imagine that labs were not trying to figure this out.
We were in distinct minority, indeed, yes.
And then met with skepticism. And
some skepticism.
Yes.
But the results are simple.
Yes.
You can reproduce it. But regarding the original diffusion chamber that we put together, it is still used, so we licensed that technology to Novabiotic Pharmaceuticals.
Okay.
It's a startup biotech company in Cambridge, not far from here. And the initial diffusion chamber is still working.
Okay.
It's still working. But since then, we came up with a number of modifications. And I think what you might have seen both in our paper and the press is a different gadget.
Okay.
So let me tell you of the two other innovations that we came up with and that Novabiotic has been using. So one was a trap. We call it an actinomycete trap. So as I mentioned, Selman Waxman was focusing on actinomycetes and showed that they are the most prolific producers of antibiotics. And we thought that it may be useful in a perfect world to have a diffusion chamber that only grows actinomycetes and nothing else, right? And so we came up with the idea of a trap, and that is that, and that really was based on the ability, unique ability of actinomycetes to form hyphae, which are long thread-like appendices, just like fungi do, and crawl through solid media and through small pores. And so what we did, we took a slab of solid agar, sandwiched it between two semi-permeable membranes and made a diffusion chamber, but didn't inoculate it with anything. And that empty, sterile diffusion chamber went onto the soil. And actinomycetes were the only ones who were able to crawl into it. So it's essentially a lobster trap idea.
Yes.
Right?
For little teeny-weeny things, yes.
Right. And so then, after a while, they crawl in and form colonies, and now you have a trap that only grows actinomycetes. So that was one thing that was developed in my lab, and then it moved to Novabiotic. And then Slava in his lab developed what you probably heard of as an iChip.
Yes.
So Slava decided to take the idea of the diffusion chamber and make sort of a massively parallel variant of that for two reasons. One, just to have more of the same in a small gadget, but more importantly, to simultaneously isolate and culture an organism. And that is achieved in the iChip, which is a little plastic platform with a lot of holes. And if you take that platform and dip it in a suspension of bacteria, each of those holes or wells captures a droplet with one cell, approximately one cell, right? So now you've automatically isolated it. You don't have to worry about purifying this organism. Then forms a pure colony, right? And if you have a lot of these mini chambers, then of course you speed up.
Right.
Isolation.
Right.
So that was the iChip, and that's, it has been also used by Novabiotic with quite some success.
I see. So, first of all, let me get straight. In a sense, you can farm, so to speak, because one of the problems in the lab was they grow so slow. But this doesn't grow so slow. You're sort of scooping them in.
They don't. Uncultured bacteria do not grow in the lab at all. They don't grow on a petri dish.
All right. So
at
it has to be okay.
We grow them in their natural environment.
Okay. Right. But you can get abundance.
So once they did grow in the diffusion chamber, in the trap, then of course a big problem is how to move them into the lab.
Tempt them into the lab.
So what we discovered together with Slava is that once a cell forms a colony in the diffusion chamber, then with high probability it will also grow on a petri dish.
Okay.
We call that domestication. We do not quite understand the nature of that phenomenon, but it has been obviously very useful. So it looks like the barrier, the real barrier, is for a single cell to form the first population. Once it did that, then it will also grow on a petri dish.
Okay. Can you tell us in a way that we can understand how it works given a bacterium that is the target that it's going to attack? How it works? Are there different ways in which these selected microbes attack a germ bacterium?
Right. So in the soil, which is a very densely packed environment, you have about 1 billion cells per gram of soil. They, of course, fight each other.
Right.
And they fight each other with antibiotics. And then we borrow those weapons to fight our pathogens.
Right.
And so we essentially, so the next step, once we have uncultured bacteria growing on a petri dish, then we emulate Selman-Waxman's method. So we take a colony of a potential producer of antibiotic, we plug it in the center of a petri dish, which is seeded with something like staph. And if there's a zone of inhibition of staph growth around the producing colony, we know that it's making an antibiotic.
Okay.
And then we can grow up this producer, extract and separate into fractions, separate that stuff that it's been making, and purify the active ingredient, which is going to be an antibiotic. And so that work has been conducted primarily by Novabiotic.
Okay.
And to date, they discovered 25 new compounds, of which the latest and perhaps the most exciting has been teixobactin. And that's what they, my lab, and our collaborators have been focusing on.
Right. What's so great about teixobactin, seeing as you've got an array here of possibilities, what and how does it work?
Right. So one thing that we've been looking for is, of course, an antibiotic to which there will be low probability of resistance development.
Yes.
Right. And so, we have a very simple test to do that. If you isolated a new antimicrobial compound, a new antibiotic, you put it in a petri dish, and then you seed onto that petri dish a large number of cells of a pathogen, and you look for resistant colonies to show up. And then you divide the number of resistance colonies by the number of cells that you seeded, and that gives you the probability of mutation, right? So it's very simple arithmetic.
Right.
And so we did that in my lab with staph and tuberculosis, and at Novabiotic. There have been extensive testing against staph, and we couldn't find any resistant mutants.
Right.
And usually that is not a good thing, actually, to find no resistant mutants. So what you are normally looking for, you're looking for a low probability of resistance.
Oh, I see.
High probability is obviously bad. That's a no-go.
Right.
No resistance is usually also a no-go, because that means that you discovered a new detergent. Bacteria like to make detergents, stuff that we put, in laundry wash. And detergents destroy membranes, both bacterial membranes and our membranes, right? There's no specific targets, there's nothing to mutate, there's no resistance development. So that's just sort of a boring proposition. But we have been on a lookout for interesting compounds with very low resistance. And so in parallel, these have been tested against mammalian cells for toxicity, right? So a detergent, of course, is toxic against mammalian cells. This compound had no toxicity, no measurable toxicity against mammalian cells. And that's when we sort of set up and
got all excited, right?
Yes,
so did everybody else, right?
Well, not at that point. At that point, a small number of people got excited, right, at that point. Because we felt that we have something very interesting in our hands. And at that point, so Lucy Link at Anomaly ran a very straightforward test. And the test was to see which important process in the cell this antibiotic is inhibiting. And by her test, she found that it's inhibition of cell wall biosynthesis.
Yes, that's what the diagram.
And that's also what penicillin targets, that general process, although by different mechanisms. And so then we reasoned that, okay, so now we have a compound. We know that it's specific, it targets a specific function in the cell. There are no resistant mutants. And that suggests to us that it does not bind or inhibit a protein target. Most antibiotics hit proteins, that have important functions in the cell. Proteins like the ribosome that makes other proteins, the DNA polymerase, RNA polymerase, and so on. And of course, proteins are coded by genes, and those can mutate. And the most common mechanism of antibiotic resistance is when a protein changes in a way that it no longer binds an antibiotic.
Okay.
Right. So now it becomes resistant to the antibiotic. So we figured no mutations should be something which is not a protein. And what we know about the cell wall, it's made of two major polymers, peptidoglycan and teichoic acid. Those polymers are not proteins, so if our compound binds to those, then there's not going to be any
you're home free, so to speak,
by mutations. And so we, at that point, we decided to involve Tanya Schneider's group. So Tanya Schneider is an expert in those precursors of peptidoglycan and teichoic acid polymers, right? She's a biochemist. That's what she does for a living. And so Tanya gave us a little bit of the precursor, and I tested that in my lab. It was a very simple test. We add teixobactin to staph, and it inhibits their growth. And then we add that precursor, which is called lipid 2, to the suspension of cells, add teixobactin again, and there's no effect. That means that teixobactin binds to lipid 2 that we added.
I see.
Right. That means that is what is the target of teixobactin, lipid 2. So that very simple experiment was followed on by Tanya's group with a number of very sophisticated examinations where she found that is indeed the case, confirmed our initial finding. But also she found that it binds this precursor of teichoic acid. So now there are two targets, right? They're somewhat similar but distinct. So it binds two targets, none of which is mutable.
Right. Both of this involves the cell wall. Just
That's
wanted
correct.
to make sure that we understand
These two targets are precursors of cell wall polymers.
Right.
And there's another very important twist to this story, which is relevant to resistance. Bacteria have a number of resistance mechanisms. Some of them are highly sophisticated, like there's a whole biochemical pathway that modifies the target, right? But usually these highly sophisticated mechanisms come from an organism that produces the antibiotic. That kind of makes sense. If you're making an antibiotic, of course you need to protect yourself, right? And the simplest way is to protect your target, to modify it. And so we were wondering what's going on with the producer. Now, the producer is a very interesting organism. First of all, it is not an actinomycete, as producers of most antibiotics are. It belongs to a very different group. It's a beta proteobacterium, and it is a gram-negative organism, and that is important.
Yes.
So it came from not from a terribly exotic location. It came from a grassy field in Maine, where one of the Novobiotic employees was sampling soil, and then that bacteria seemed pretty interesting and unusual. And Amy Sporing, a scientist at Novobiotic, picked it up. So she discovered this bacterium and felt that it's interesting enough to follow through. So gram-negative bacteria have a second membrane. All cells will have a cytoplasmic membrane.
Right.
Gram-negative bacteria have a second membrane encircling the cell. And that second membrane has a very simple function: to prevent penetration of antibiotics.
That's why that's so resistant.
Right. So what did this be? So now this bacterium that's making teixobactin, it protects itself from this compound by exporting it across its outer membrane. It doesn't have any specific mechanism of resistance. It just throws it out. And since it cannot come back, that's the resistance mechanism.
That's amazing.
So for a gram-positive bacteria that it hits, there's nothing to borrow. There's no mechanism. They cannot borrow an outer membrane, right?
I see.
It's not feasible. So it looks like this compound, teixobactin, has this multi-layered protection from resistance. We've never seen anything like that.
I was going to say this is a real minority here.
So it's clearly it evolved to be largely free from resistance, right? It's not an accident. But that also tells us that that is obviously not the only compound like that out there, right? We only scratched the surface, literally, of the planet.
And so you think it's not necessarily so rare as it would look like at the moment that you've discovered it. There may be others that have evolved this kind of mechanism.
Absolutely. I'd be astonished if we, with a fairly modest effort, between our academic labs and Novobiotic, in the enormous planet Earth, happened to hit the one and only
and all that
compound.
Soil bacteria.
Right. The enormous number of species.
Right.
Of course, this is not the one and only compound that is largely free of resistance.
Well, does that mean then that that is actually a leap in antibiotic research, that you're looking now in a different direction altogether?
Well, yes.
Because you've discovered this mechanism?
I would say yes. And you see, we've all been operating under the standard dogma in the field. And our standard dogma is that bacteria will always develop resistance.
Yeah.
And so then you have really one option. And your one option is to discover compounds faster than resistance is going to develop.
Right.
People have been advocating also another, if not approach, but very useful thing, and that's judicious use of antibiotics, good stewardship. And that's, of course, absolutely necessary. But in reality, we can introduce such things in Europe, in America, but it is considerably harder, especially in short term, to introduce such things in third world countries where antibiotics are going to be sold over the counter for the foreseeable future. And so really then our only option would have been to discover new antibiotics faster when pathogens acquire resistance. And now we have another option, which we have not really thought about as being realistic. And the option is to discover compounds with very limited propensity to resistance development.
It's really quite amazing. And this is effective as far as you know against, you mentioned tuberculosis, which has been a disaster because it's coming back now and it resists everything you throw at it. The other one is MRSA, this awful thing that we associate, I guess, with hospitals now that it's
and also with community.
I see.
MRSA is
yeah,
also in community.
But a number of these things where you used to be able to take care of infections and diseases that were bacteria, suddenly they loom as really ominous. But this, it looks like, because it's a different avenue, it looks like it's highly effective.
Yeah, well, it is effective, and it's effective in mice. So it cures mice of a number of difficult to treat infections. But of course, it's not yet in the clinic.
That's the next thing. Because just to remind viewers that there are always concerned that mice are not human, and so scientists can't run around doing this, testing these things on humans prematurely. What are the steps, and what is the length of time?
So the steps are, first of all, more toxicity studies.
Yeah.
And more studies on efficacy in additional animal models. And then this compound, teixobactin, could enter into human clinical trials. But it also has limited solubility. We would like to see a higher solubility compound where we can deliver, especially in humans, if needed, a higher therapeutic dose.
I see.
So Novobiotic now is taking on a medicinal chemistry approach to get analogs of teixobactin with improved solubility. So these are sort of two parallel paths.
I'm not clear on that. Is that in order to determine what dosage? Is that what it is that works with different diseases?
So it has limited solubility, right? And we could cure a mouse. And as you said, a human is not a mouse. Now imagine that humans need a larger dose, right? If humans need a larger dose, we need a more soluble compound.
I see.
So we can deliver that larger dose, right? So we need to be prepared for that eventuality.
Right, I see. This looks very exciting. Is this it had to be a big thrill for you and your lab and your colleagues.
Yes.
I think it's amazing just because it's a great leap forward, so to speak. It seems like a great leap forward. What do you predict for the future, apart from developing this, which seems to be, a given? But the other is, it sounds as though there's a whole new track for microbiology now, that people will be doing things in a different way. What is the future?
Yeah. So teixobactin has a very complex mechanism of action. The reality today is that it is very difficult to come up with a compound like that using synthetic chemistry. Right? We're not there yet. So I think in the short term, in the next years, at least not decades, but years, probably the best path forward, capitalizing on this discovery, is to broaden the search for new exciting teixobactins, right? For new exciting antibiotics, using the platform that Novobiotic has been exploiting, the uncultured bacteria. And that is what hopefully we'll be able to do to enlarge that effort. Ultimately, of course, we people will have to learn how to synthesize terrific antibiotics. We're just not there yet.
But you do anticipate that they will synthesize this, that they...
Well, of course. There's no reason we shouldn't be able to. It's just the state of the art in chemistry and rational drug design is not quite there yet. But I think we'll get there.
It's really thrilling a moment. I think it must be tremendously gratifying at just this moment when people were throwing up their hands about bacteria, about these bacterial infections, that you've come up with a fabulous discovery. So I think it must be very exciting on your end.
Yeah, sure. Well, to be fair, not everybody was throwing up their hands. I mean, there's quite a number of labs and groups and companies that are working successfully on the problem. I think what we've contributed rather uniquely to the problem is to show that there is a method, not just a single compound, but there is a method that can be exploited, this discovery platform, that will allow us to discover additional compounds.
I see. Does this entail also retraining people who are going into microbiology, looking into this sort of thing? Or is this just something new?
I don't think it's fairly simple. As we said in the beginning of this conversation, the question was, it's so simple, why didn't other people think of it? So I don't think this requires a lot of training.
Okay, all right.
So
it will be a different avenue in any case. I would like to point out before we close here that you've done some really exciting work on Lyme disease and a number of other things that I hope to pursue at another point, but that your lab is really something that I hope, because of the recent publicity, will become much better known to the general public now. But I thank you very much for joining us today and giving us this wonderful discussion. Thank you.
Thank you, Ron. Thanks for having me.